Virgin birth among animals may not be a rare last resort save the species stopgap after all. For the first time, animal mothers, specifically pit vipers, have been discovered spawning fatherless offspring in the wild. Remarkably, the snakes did so even when perfectly good males were around. Among vertebrates that typically reproduce sexually, virgin birth or parthenogenesis had been observed only in captive female snakes, Komodo dragons, birds and sharks. Until now, parthenogenesis was considered an evolutionary novelty, a way for a bloodline to continue in the absence of suitable fathers. In a study published in this week's issue of the journal Biology Letters, a team led by biologist Warren Booth of the University of Tulsa in Oklahoma captured pregnant copperhead and cottonmouth females from fields where males were present. When the snakes gave birth, the researchers documented the physical and genetic characteristics of the litters. Tests showed that one of the 22 copperhead mothers and one of the 37 cottonmouth snakes had given birth parthenogenetically, a ratio Booth finds surprisingly high. The fact that we find it in such small sample sizes is quite remarkable, Booth said. What we're going to do now is go back to these populations and do sampling year to year to see if we can find instances of parthenogenesis again. Why would female snakes undergo parthenogenesis when males are available? One possibility is that it might have been the only way they could reproduce. Booth noted that the copperhead that underwent parthenogenesis was smaller than usual, and perhaps passed over by males in favor of fitter females. Other scientists suggest parthenogenesis might be a random reproductive mistake. Booth himself is investigating whether a bacteria or virus triggers it. Another mystery is that each of the 2 virgin birth litters consisted of a single snake. A normal copperhead litter might number between 6 and 9, while cottonmouths can spawn up to 20 offspring. Also, the 2 virgin birth baby snakes were both male. Interestingly, every known parthenogenetic snake offspring has been male. It would be interesting to see if we can find females, Booth said. There's no reason why we shouldn't find females, but in all the snake species we've looked at, they've all been males. It's also unknown if parthenogenetic animals can reproduce normally or have virgin births themselves. Parthenogenetic offspring often exhibit abnormalities or die early, which Booth says is essentially a severe form of inbreeding. A virgin birth occurs when a polar body, a cell produced along with the egg, functions like a sperm and fertilizes the egg. Consequently, the DNA of a virgin birth offspring, or parthenogen, doesn't perfectly match that of its parent. The offspring is a sort of half-clone. So far, parthenogenesis has only been observed among sharks, reptiles and birds, which are closely related to reptiles. Mammals aren't thought to be capable of parthenogenesis because their reproduction requires copies of genes from both parents. So no human parthenogenesis anytime soon, said Stony Brook University marine biologist Demian Chapman, who discovered virgin birth among black-tip sharks. We'll leave that to the snakes, birds and sharks.